So, welcome to study party number 2 of the 2012 Christmas exam. This is recording number 1. We are going to start with chapter 5 on page 74. So, this first section is talking about the fluid mosaic model of the cell membrane. And if you look at figure 5.1a, what you see is that figure is essentially the same figure that you saw in chapter 4 when we were looking at cell membranes. And so you see the phospholipid bilayer. Okay? You see the cholesterol molecules embedded in the center. You see proteins, colored purple. Some proteins are surface proteins on the extracellular surface. Other proteins are intracellular surface proteins. And other proteins are called integrins because they go all the way through the membrane. On the inside of the membrane, we have the microfilaments, which are made out of actin, remember from last chapter. Okay, that supports and gives structure to the cell membrane. And on the outside, we have glycoproteins, okay, which are like carbohydrates stuck onto the proteins. We have glycolipids. Often those work as identifiers. Okay. Now, remember today in class we were looking at histone proteins, and we one of the comments, I think actually Kelly might have made it, was that, oh my gosh, this textbook like so oversimplified the drawing. Okay, what you're not seeing on this is the actual structure. Look at those enzymes, those proteins. They're purple blobs, right? We know better. And so some of those proteins could be receptors. And you see that in figure 1.c. Some of those proteins could be enzymes, running enzyme reactions on the inside of the membrane. Some of them could be transport proteins. And so those enzymes do lots of different things. Okay? So they can support, they can do cell-to-cell recognition, they make junctions between cells, so they're only showing you one cell here, not showing you a junction. They could be enzymes, they can do signal transduction, they can do transport. Okay, so those enzymes, those proteins in the cell membrane can do lots of things. Now remember that it's called a fluid mosaic. Fluid, it has sort of the consistency of salad oil. And so those proteins can float around. Remember our analogy of putting all the ping pong balls in the surface of the pool and then throwing in some inner tubes? Okay, stuff can move around. Right? Now, membranes tend to form spontaneously. They don't need anything special because of their hydrophilic interactions. So when you drop some oil in, it forms a little round blob. Okay? And so the fact that membranes form spontaneously was probably a really important step because, once again, membranes provide separation. It provides places where stuff is and places where stuff isn't. But that being said, even though we're providing compartmentalization and separation, we still have to get things in to the insides and out to the outside. So we still have to move stuff across this membrane. But we don't want to let just any old thing across the membrane, right? And so when things move, they tend to move down their concentration gradient. They move from high concentrations to low concentrations. And if they're just moving, we have diffusion. Okay? So diffusion, just moving from high concentration to low concentration until we reach equilibrium. At equilibrium, it doesn't mean that there's no net movement. It just means that the movement, or it does mean that there's no net movement. It means there is still movement, but the net movement is zero. Okay? Now, if it's moving across a membrane, we call it passive transport. Okay? Much of the really important stuff that goes on in our body happens because of passive transport. Okay? Oxygen diffuses across into our lungs, CO2 diffuses out, water moves, okay? And it's all based, again, on this concentration gradient. Now remember, in figure 5.3a, we're looking at passive transport of just one type of molecule. And then if you look at figure 5.3b on the bottom of page 75, we're looking at passive transport of two types of molecules. those two types of molecules move down their own concentration gradient. Not the constant, it's as if the other molecules didn't even exist. Okay. On page 76, section 5.4, osmosis, a special case of diffusion. And so remember that the full and correct definition of osmosis is the diffusion of water across a semi-permeable membrane. And so you have to have diffusion, you have to have water, you have to have a semi-permeable membrane. So osmosis only refers to the movement of water. So we have to pay attention to where the water concentration is the greatest. And so in this diagram, 5.4, you can see that when there's a barrier and there's a difference, there's a higher concentration of solute, that means there's a lower concentration of water. And so water moves to that higher side, and you can see that we get a differential height of the water in that U-shaped tube. Okay? So far, so good? Okay. Now, water balance is critical for your cells, for you as an organism. Okay? And so we have three words that refer to the solution, not to the cell. We have isotonic, which means the concentration inside is this equal to the concentration outside. And remember, once again, I'm talking about the concentration of water, not the concentration of anything else. Okay? So isotonic means the water concentration is the same inside as outside. Hypotonic means that there is more water outside than inside. So which way does the water move? The water would move in, the cell would swell. Now, if hypotonic. Oh, so I think hypo with an O, the cell swells up like a big fat O. If it's an animal cell, it will burst or lyse. If it's a plant cell, it takes in all of that water into its central vacuole, and the cell becomes turgid. The cell wall keeps the plant cell from bursting. The opposite of hypotonic is hypertonic. And I think about hyper, the cell sort of shriveling up like an E. Okay? And that's because the water concentration inside the cell is greater than outside the cell. So the net movement of water will be out. The cell will shrivel if it's an animal cell. If it's a plant cell, it'll still shrivel, the plant wilts. Eventually, you have plasmolysis occurring where the plasma membrane pulls away from the cell wall. That's often in terms of the whole organism of the plant. Sometimes that's referred to as the permanent wilting point. So, remember this summer, you forgot to water your coleus. It wilted. Oh no, you said, and you raced back in and you gave it some water and it perked right back up. Oh, phew, right? Had you let it go past its permanent wilting point and plasmolysis had occurred, it wouldn't have recovered. And perhaps some of you noticed that maybe a leaf or two of your plant never did fully recover. It just sort of died and fell off. That's because plasmolysis occurred. Okay? Not because you put it in a hypertonic solution, but because... Sorry, little blanket malfunction for those of you not here this evening. But Marley's okay now. Phew. All right. So, moving on. Section 5.6. So, not everything can move across the cell membrane by diffusion. And so we have to have facilitated diffusion. In facilitated diffusion, we have to use one of those transport or channel proteins. And so in figure 5.6, at the bottom of 77, you can see that transport protein forming a channel that solutes can move across. Those solutes are going to move from high concentration to low concentration. Now, sugars, amino acids, ions, even water uses actual facilitated diffusion. The protein that helps water move across the membrane through facilitated diffusion is called an aquaporin. Peter Augury won the Nobel Prize for that in 2003. And in section 5.7, he talks about his discovery of aquaporins through his study of kidney cells, okay, and looking at blood. In the next section, 5.8, some molecules are just too big to get across. Other molecules, we want to move against their concentration gradient. In both those cases, we need active transport. We need the expenditure of energy to move those molecules, either because they're too big or we want to pump them back against their concentration gradient. And so active transport requires some form of energy, often in the form of ATP. And in diagram 5.8, you see the four-step process. So the solute binds almost to an active site, but it's not an enzyme. ATP comes along and phosphorylates it, so ATP drops off a phosphate, the molecule changes shape, transports those solute molecules across to the other side. As soon as the phosphate functional group detaches, the molecule returns to its original shape. And so this works like a pump, pumping molecules across the membrane. Okay? Now, if the molecules are really big, they're way too big to get pumped across with a transport, we have to do things like exocytosis or endocytosis. Okay? Now, depending on scale and how big the molecules are, exo and endocytosis have different names. So phagocytosis is sometimes referred to as cell eating, in which we're engulfing very large particles, as you can see at the top of figure 5.9. Okay? So we engulf and we create a food vacuole. Remember, anytime we do exocytosis or endocytosis and a vacuole or a vesicle either forms or fuses with the plasma membrane, remember that the insides of the membrane become the outsides. And if you remember, we actually modeled that in the pod with the little flags, how the insides become the outsides. That's really actually very important when we get to cell signaling. And so, for example, insulin causing glutes to move from the inside to the outside to facilitate the uptake of glucose. If the molecules are smaller, we don't need to do phagocytosis. We can do penocytosis, which is often considered cell drinking or gulping. And so instead of putting those big particles into food vacuoles, the engulfing process is similar, but we end up with a little vesicle. Okay? And then we have receptor-mediated endocytosis. Very, very, very specific. Phagocytosis and penocytosis are more generalized. We're just engulfing whatever comes along. But in receptor-mediated endocytosis, what's happening is that there are receptors, usually proteins, embedded on the outside of the membrane. When a specific molecule binds to that receptor, it causes then this little coated pit to form, and we end up with a coated vesicle. Okay? For example, this happens in your body when we take cholesterol from the blood in order to make new membranes. Okay? So, cholesterol circulates in the blood. We have LDLs and HDLs, remember? And which ones are good? The low-density cholesterols, lipoproteins, or the high-density? Low. We want more low density. And they bind to receptor proteins and then enter the cell by endocytosis. And then we can use them, okay, to make more membranes. Now, some people have this inherited genetic disorder called hypercholesterolemia. And they don't have enough LDL receptors. And so they can't absorb through endocytosis, receptor mediated. They can't get all of that cholesterol into their cells, so the cholesterol stays in their blood, and they end up with really high cholesterol. Okay? So that takes us to the end of this section of the chapter about cell membranes, osmosis, and diffusion. The one piece I didn't really talk about in this section, because it's not in your book, but remember it is in your lab, is calculating water potential. And water potential is the total of the solute potential plus the pressure potential. Water moves from high water potential to low water potential. Okay? And so remember the symbol for water potential, the Greek symbol phi. Okay? And so that might be something.
It moves from high potential to low potential?
Right. So
and it's total of solute.
it's the solute potential plus the pressure potential. Because let's say I have a bag of sucrose and water. I have a dialysis bag and I put it in a solution of pure water. Is that solution hypotonic or hypertonic or isotonic? Hypotonic, because water's going to want to move in. And so water's going to continue to move in because the solute potential is very high. Okay? But eventually enough water is going to move in, that little sausage of dialysis tubing gets as full as it can possibly be. There's no more room to squeeze in any water molecules, and so the pressure gets really high. And then the pressure, one molecule moves in, but there's so much pressure it pushes a molecule out, even though it really wants more water to come in, right? So it'll never reach equilibrium. Okay? So does this sort of ring a bell a little bit? Okay, so I'm going to stop the recording now and take any questions that you might have. So, welcome to study party number 2 of the 2012 Christmas exam. This is recording 2, and we're going to start on page 80, chapter 5, section 10. Okay? We just finished talking about active transport and how cells might need energy to pump things against a concentration gradient. Well, we need all kinds of energy to do all kinds of stuff. Pumping things against their concentration gradient is just one example. Okay? So, we know that we require energy to stay alive. And we also know that energy comes in various forms. We have kinetic energy, the energy of motion. We have potential energy, which is stored. Okay? In both those cases, whether it's kinetic energy or whether it's potential, it's the capacity to perform work, the capacity to do something. Okay? Now, some types of potential energy include chemical energy, the energy that's locked up in chemical bonds. Okay? Now, all energy has to obey the laws of thermodynamics. The first law of thermodynamics is the law of conservation of energy. The sum total of energy in the universe stays the same. Energy can change forms, but the sum total of energy remains the same. Okay? It's very similar to the law of conservation of mass. Now, if this were a physics class, we'd have to have all kinds of caveats because we know that E equals mc squared and matter and energy is interchangeable, and thank goodness for that, or we wouldn't have the sun, right? But for our general purposes, the main principle is that the energy is conserved, okay? The second law of thermodynamics is the one that gets us, though. And actually, it's the one we were sort of grousing about at the beginning of the study party before we started recording. And that's the law of second law of thermodynamics, or the law of entropy. Entropy is a measure of disorder. Things always move from order to disorder. My life is a classic example right now of entropy winning. Entropy tends to win. The only way to combat entropy and to create order out of disorder is to input energy into the system. So, if your backpack looks like it's exploding, entropy's winning. If your car has the remains of the entire second quarter's lunches tossed carelessly in the back because you were going to be late to your afternoon class, entropy's winning. If you have to shuffle through your room lest you step on something important and break it, entropy's winning. Look at all these nods of recognition. So, in order to regain order, you have to put energy into the system. You have to put energy into organizing your backpack, cleaning up your room, emptying all the trash out of your car. because it's starting to smell bad, whatever. It takes energy. And what often happens to us, okay, so this really isn't a biology lecture, this is the motherly lecture leaking in, okay? We often say, oh, we don't have time to do that because we have all of these other pressing things. If we actively combat the entropy of our lives, we often find that we have more energy and more time than we thought we did. That half hour you spent looking for your car keys this morning? Under the pile of stuff in your bed? Yeah. Well, with just a tiny bit of energy the night before, taking that 30 seconds to put them where they belong would have saved you then 29 minutes and 30 seconds this morning, right? So, fighting entropy is a noble and just cause. Dun da da da. End of that motherly lecture. So, moving on, but check this out. I love this diagram on 511. In our car, we put in gasoline and oxygen. Gasoline holds an enormous amount of potential chemical energy. When we burn it and chemically combine it with oxygen, we see that heat energy is given off. That's the less useful energy. That's the entropy winning. And we have kinetic energy of movement, the waste products, carbon dioxide, and water. Cells do the same thing, only we're going to take glucose and oxygen, and essentially we're going to burn it. We lose it as heat, that's entropy winning, and we actually produce ATP to save to do other types of cellular work. So we transform, really what we're doing is transforming one kind of chemical energy into another kind of chemical energy that is more useful. A byproduct, again, being carbon dioxide and water. However, when we actually measure energy efficiency, the energy efficiency of a cell is like orders of magnitude higher than the efficiency of your car. Okay? Because, unfortunately, as always, we cannot replicate the efficiency of biological systems. Okay? So, when we have our chemical reactions in section 5.12, they're either going to release energy or they're going to store it. So, a chemical reaction that releases energy is called exergonic. Think exit. Okay? A chemical reaction that requires energy to run is called endergonic. Ender, thinking enter, it has to take energy in. All right? And so, metabolism is the sum total of all of the reactions in your body that either require energy to run or give off energy. Okay? Now, think about this. Where could a reaction that requires energy get the energy to make it happen? From a reaction that gave off energy. And so what we often find are what are called coupled reactions. We're going to couple a reaction that gives off energy with a reaction that requires energy. So we can use the energy that's given off to drive the next reaction. Okay? So when we're talking about coupled reactions, that's what we're talking about. Okay? On page 83 in section 5, oh, sorry, yes, go ahead.
Sorry, just really quick. So a coupled reaction will always be an exergonic and endergonic combined?
In general, yes, because it takes the energy released from one to drive the other.
Could there be like coupled or multiple where there's more than one exergonic and all that energy is combined to?
Oh, so where two exergonic reactions have to release enough energy to create the one ender one. Sure, absolutely. Okay, can I think of a specific example to tell you right now? No. But yes, that's very possible. Okay, and so what do? We have a coupled reaction in the creation of ATP. Okay, so we use an exergonic reaction we take the energy from an exergonic reaction and we use it to endergonically put on the phosphate group, that third phosphate group, onto an ADP. And so, if you look at figure 513A, ATP is a nucleic acid. How can you tell? Because there's an adenine, which is a nitrogenous base. It happens to be a purine. I can tell because it has 2 rings. Big molecule, little word. It has a pentose sugar, ribose. And instead of 1 phosphate group, in which case it'd just be an adenine base, it has 3 phosphate groups. So adeno, adenine, the aden from adenine, the ose from ribose, tri for 3 phosphate, adenosine triphosphate. We also, even though your book doesn't tell you, you can also have GTP reactions. Guanosine triphosphate. Okay, and it works just like adenine ATP, adenosine triphosphate. Okay, and so we have hydrolysis. It breaks off that 3rd phosphate, releasing energy in an exergonic reaction. So it takes energy to make it, and we will look more specifically at how ATP is made when we come back and look at chapter 6 and cellular respiration. All right, so I'm going to stop here because this recording is getting a little long. In our next recording, we'll talk about enzymes and how they work to lower the activation energy. Oh, there we go. This is recording number 3 of study party number 2 for the Christmas exam 2012. We're going to start with, oops, I turned the wrong page. We're going to start on page 84 with section 514, looking at enzymes. Okay, enzymes are, in general, proteins. Okay, they are proteins that serve a specialized function. So, enzymes work to speed up reactions, and so therefore they're biological catalysts. A catalyst does not get used up, but it facilitates a chemical reaction happening. The reason that an enzyme speeds up a chemical reaction is that it lowers the activation energy of a reaction. So if you look at figure 5.14, a reaction without an enzyme, there's always this little activation energy. So if you think about a ginormous boulder at the top of a hill, okay? There is enormous potential energy up there, right? And all that boulder needs to do is it just needs a little push. And if I gave it just a little push, that boulder is going to lose all of its potential energy, and it's going to turn into kinetic energy as it rolls down the hill and releases all of its energy, crashing into the bottom, crashing into the highway. Okay? But the boulder's just sitting up there. Why doesn't it roll down and crash into the highway? Because it needs that little push. That little push is the activation energy.
So enzymes lower that?
So enzymes lower that. Okay? Does that make sense?
So instead of a push, it would be just
Yeah, so instead of a big push, and maybe taking all of us to push that boulder, or using a lever or something, you could just blow on it maybe, and it would fall down. So if all I had to do was just tap the boulder and it would fall down, as opposed to using a lever and all my force, I could make a lot more reactions happen just by tapping boulders. Okay, so I can speed up that process considerably. Okay, and so notice that the reactant has a higher energy than the product, and so is this an exergonic or an endergonic reaction that I'm showing you?
exergonic.
In this diagram, it's an exergonic because the reactants have more energy than the products. So energy had to be given off in that process. Okay? Now, enzymes are highly, highly, highly specific. And the reason that they're very specific is they have a specific 3-dimensional shape. Why do they have a 3-dimensional shape? Because they're proteins, and they have a primary structure, which is the order of amino acids. They have a secondary structure, which is an alpha helix or a beta pleated sheet. They have a tertiary structure, which is following the rules of folding that we talked about at last night's study party, right? And so we're going to have this globular protein, tertiary or quaternary level structure, and it's going to have places that stick out, and it's going to have places that stick in. Lots of them. Okay? A place that sticks in, that the substrate molecule is going to fit in, is called the active site. Now, if you think about it, this globular protein is, 30 to 300 amino acids long. The active site is generally formed by 8 to 10 amino acids. So what do we need the rest of the amino acids for that aren't forming the active site? The rest of the structure for stabilizing the molecule, for stabilizing that active site. Okay? So they play a very important part. So in figure 5.15, down at the bottom of page 84, in this case we see that the substrate is sucrose, so therefore it's a carbohydrate because it ends in -ose. The substrate, or the enzyme, is often named after the substrate that it does catalysis on. So the enzyme that breaks down sucrose, sucrase. You can always recognize an enzyme because it ends in ase. Okay? Sucrose binds to the substrate with an induced fit. That means it's not a perfect fit. Okay? So it adds some torsional pressure, and that torsional pressure moves sucrose to its transitional state. This is all fancy chemistry talk, right? It moves it into its transitional state, and then that's the push to cause it to break apart into glucose and fructose. Once it breaks apart, it comes out of the active site, and the enzyme is back just the way it was. Okay? Now, remember that enzymes are highly, highly specific. Enzymes are proteins, and therefore they have very specific environmental requirements. And so there are optimal conditions. There's an optimal temperature. There's an optimal pH. There's an optimal salinity. If the pH is too high or too low, or the temperature is too high or too low, the rate of reaction slows. Okay? And the same thing is true for salinity. And so if the rate of reaction if the temperature, for example, gets too high, remember that temperature is a measure of heat. Heat's a measure of the kinetic energy of the molecule. Okay? So if the temperature gets too high and that vibrational kinetic energy gets too big, the tertiary level structure can't hold anymore. The enzyme falls apart. The enzyme is denatured. Generally, denaturing an enzyme is a sort of one-way process. You can't undenature it. When you fry your egg white, you denature the albumin protein, and you can't go from the hard white, rubbery egg white back to the gelatinous egg white that you had when you first broke the egg white, right? So denaturization is generally a one-way street.
Is that always what happens when you cook something?
Yes, in general, what you're doing is you're denaturing proteins. Okay? You can't go from cooked meat back to raw meat. You can't uncook your meat.
Where does denaturing of enzymes
Mm-hmm.
in like a cell or something,
Mm-hmm.
if it is denatured, are those amino acids reused?
Right. So the amino acids are still intact. They still have their structural integrity. So if the enzyme is no longer functional for whatever reason, then we're going to probably engulf it in a lysosome, use protease enzymes to break it apart. Okay? And then we're going to do hydrolysis, break it down to its individual amino acids, which then we can recycle into a new enzyme when the next time we do transcription translation. Okay? So we just keep breaking it down. Now, some enzymes work best if they have a cofactor. Okay? That cofactor binds to not the active site, but a different place on the enzyme and makes the active site be the perfect shape. Now, If the cofactors are inorganic, they could be like zinc or iron or copper or magnesium, okay? Then it's just called a cofactor. On the other hand, some cofactors are organic molecules. And then instead of a cofactor, we call it a coenzyme. Vitamin C, for example, is an organic molecule that behaves as a coenzyme. Okay? Now, we can have enzyme inhibition. It blocks the enzyme action, and we can control whether the enzyme works or not. We can have competitive inhibitors, where the inhibitor actually lodges itself in the active site so that the substrate can't fit. We can have a non-competitive inhibitor where the inhibitor binds to a different place. But when it binds to a different place, the non-active site, it actually changes the shape of the enzyme again so that the substrate doesn't fit. So in both cases, the substrate doesn't fit. Okay? Penicillin is a really good example. They inhibit the enzymes of bacteria. So when you take an antibiotic, you're inhibiting the enzymes of a bacteria which are different than yours, right? Because it's prokaryote. And then you're killing that bacteria but not killing you. Yeah.
So are non-competitive permanent and competitive are not permanent?
It depends. It depends. Sometimes that's true and sometimes that's not.
So a non-competitive isn't always permanent.
Right. Right. In often cases, the non-competitives are not. If we can remove it, then the enzyme will work. And if you think about it, if we insert it and remove it, we can regulate whether that enzyme's working or not working.
Yeah.
Okay? And remember, too, that we can isolate and separate things, so we could keep coenzymes away or cofactors away until we really want that enzyme to work. We could block it with an inhibitor, okay? And so we do regulate. So we tend to think about inhibitors as being permanent. Pesticides, herbicides tend to be fairly permanent, okay? But if we're regulating metabolism, we want it to be impermanent. We want it to be reversible. Okay? And so we get feedback inhibition. We start making the product, whatever the product is. The product then comes back and acts as an inhibitor because we don't need to waste that energy making more product. Okay? Because we want to conserve energy so we can win the game of life, right? That brings us to the end of Chapter 5, so I'll stop this recording, take any enzyme questions that you have. So, this is recording 4 of our second study party for the 2012 Christmas exam. We begin this on page 126, Chapter 8, Section 1. Okay. Remember we talked about surface area and volume. Cells can only get so big because surface area only increases as the square, whereas volume increases as the cube, right? And so if cells get too big, they can die of waste, they can't get rid of waste, they can't get nutrients in, and the DNA can't keep up with the protein demand, so we get DNA overload, okay? But cells also divide to reproduce. And so we say like begets like, more or less. We have sexual reproduction and asexual reproduction. In asexual reproduction, 2 cells divide and we have genetically identical daughter cells. So the parent sort of dissolves into its own progeny. Okay? That's great if you happen to have a really good set of genes and you're truly well adapted for your particular environment. Divide asexually because we want to make more well-adapted organisms. But if the environment changes and you're no longer so well adapted, that's a problem. So along came the invention of sex. And the whole point of sex is to redistribute genes and to mix them up and increase genetic variability. Okay? So, we're back to cell theory. Cells only come from pre-existing cells. We don't have spontaneous creation of cells. Okay? So, The cell is the basic unit of life. Okay? And we're going to go back and revisit because prokaryotic cells are different than eukaryotic cells. And the main goal in cell division is to produce two identical daughter cells. They need to be identical because they need to have all the same genetic information if they're going to do their jobs. But because prokaryotes have circular chromosomes, they do things a little differently. They reproduce by binary fission. Binary meaning two, fission meaning to break apart. And so what happens is the prokaryotic chromosome replicates. It actually, and they don't show it in this picture, but it actually does attach to the plasma membrane. The plasma membrane grows between those two sites of attachment, pulling those two apart, and then it begins to pinch in, okay? And it ultimately divides into two daughter cells. Okay. So, interestingly, if you remember from the chapter on cells and the idea of endosymbiosis, that's how we got mitochondria and chloroplasts. Mitochondria and chloroplasts reproduce themselves within the cell. They have their own DNA. Imagine what? Guess what? It's circular. And guess how mitochondria divide? Binary fission. So we have all this interesting evidence. So in section 8.4, we're going to then look at mitosis, which is cell division of a eukaryotic cell. Remember that in eukaryotic cells, the chromosomes are linear, okay? And we talked a little bit about this today. When the DNA is all unwound, we call it chromatin. That way we can get at the A's and T's and C's and G's. We can replicate, we can do protein synthesis. But when chromatin is all unwound, it's really long, it's really big, it's really huge. And so to organize the DNA so that it doesn't get broken, it doesn't get tangled, it doesn't get damaged, and we can make sure that each cell gets its own copy, we coil it up into a chromosome. And in figure 8.4b, you see the electron micrograph of a duplicated chromosome, and you can imagine looking at it, that those are the tangled supercoils. And we'd have to do a lot of unwinding to get to the chromatin. That's two sister chromatids held together by a centromere. Okay? Lots of C word vocabulary. It's important to keep them in mind. Okay? In section 8.5, we're looking at the whole cell cycle. Figure 8.5 at the top is a really nice picture of the cell cycle. Notice that the cell is only actually dividing for a very short period of the cell cycle. The vast majority of the time, the cell is in interphase because the DNA needs to be unwound, because the DNA is the directions for making proteins. And that's a lot of what the cell does, is make proteins to carry out its metabolic processes. Interphase is divided into three sections, G1, S, and G2. G is called gap, or you could also think about it as growth. So we have the first gap, and then we have S, which is when the DNA replicates. Okay? And now, thanks to chapter 10, we know exactly the molecular process by which DNA makes an exact copy of itself. So DNA replication from chapter 10 happens during the S phase. Then we have the second gap phase, where the cell is getting ready, continues to grow, but it's building the proteins that it needs to do the cell division. Then it enters the mitotic phase. The mitotic phase consists of mitosis and cytokinesis. Okay? So, experiments. This process has unbelievable fidelity. If you made copies of CDs, if you wanted to burn 1,000 copies of a CD, you're likely to have a CD or two with a boo-boo on it that doesn't read right. Copying of DNA, way, way, way more accurate. Okay? When they did it with yeast, one error once in 100,000 cell divisions, and only one error. How crazy is that? It's remarkable. If you look on page 130 and 131, it's a really, really nice 2-page spread of the steps of cell division. What we need to do is think about the process and worry less about the names of the process. So in interphase, you can see the nucleolus, you can see the nuclear envelope, okay? You see the centrosome. The centrosome has 2 orthogonal centriole pairs. The DNA is all unwound in chromatin so that we can access the major groove, we can access the minor groove, we can separate it, we can make proteins, transcription, translation. And in prophase, the very beginning of getting ready to divide, we're going to coil up that chromatin around its histone proteins, making a nucleosome. We're going to make coils, we're going to make supercoils, we're going to compact it. Okay? And each chromosome consists of 2 sister chromatids held together by the centromere. In metaphase, those sister chromatids are going to line up on the metaphase plate. They're going to attach to spindle fibers. They don't attach with the centromere, they attach at the kinetochore. Okay? During anaphase, those spindle fibers contract, pulling the sister chromatids apart. And then in telophase, which often happens in conjunction with cytokinesis, okay, the DNA starts to unravel into chromatin. We get a cleavage furrow, so this is an animal cell, okay, and the cell divides in 2. Now our chromosomes are composed of only 1 chromatid, okay, which is going to immediately unravel. So cytokinesis in section 8.7, you can see, differs for plant and animal cells. Okay? If you're an animal cell, you get a cleavage furrow. You can't make a cleavage furrow if you're a plant cell because you've got a cell wall. So what you do is you form a whole bunch of little vesicles containing new cell wall material, which we remember as cellulose because it had alternating links of glucose. Okay? Those vesicles grow and eventually fuse, and we get a cell plate forming down the middle. Once that cell plate reaches opposite ends of the cell, we have our 2 daughter cells. Okay? Now, we don't want our cells to divide just any old time, any which way. We need to control it. Have you noticed that there's all kinds of control mechanisms, and that's sort of becoming a little theme as we get a bigger, deeper understanding of the content? We want to control when things happen, and that's really important. So one of the things that determines whether cells begin to divide or not are growth factors. Okay? Growth factors stimulate cells to divide. Okay? There's a whole bunch. We know of at least 50, probably 60 by now, different growth factors. Okay? If I get a boo-boo and I'm bleeding, platelets in my blood release factors to cause the cells that I damaged, okay, the cells surrounding the damaged area, to divide, to heal that wound. Okay? Pretty cool mechanism if you think about it. Okay?
So when you have like a lot of, actually, never mind.
Okay,
I don't know if
you
I
sure?
can start that question.
Okay. Now, we don't want cells to divide all the time. Okay? And so we have density-dependent inhibition. So when cells get too close together, when they get too crowded, they just stop dividing. Okay? The other thing that can inhibit cells from dividing is that most cells, most animal cells, have to be anchored to something. If they're not anchored, if they're just floating around, they don't divide. And so they need to be anchored to a solid surface, like the inside of a culture dish or some membrane in your body, the extracellular matrix, and then they can divide. Okay? So, in the next section, we look at those growth factors. What signals cell division? So we have this whole cell cycle control system. Okay? Notice that there are different checkpoints. There's a checkpoint, there's a really important checkpoint in G1, there's another checkpoint right at the end of G2, and there's another checkpoint in the middle of mitosis. Okay? And so, The G1, remember I said, was the most important. Otherwise, it just sort of sticks. Does anybody remember from the mitosis lab what the name of some of these factors were? Cyclins? Okay. So, it would be worth your while to go back and revisit the background information in the cell division lab. I believe that was lab 7. Okay. So how does the growth factor work? The growth factor works in figure 8.9b with a signal transduction pathway. So notice that the growth factor binds to a receptor protein. That receptor protein sends that signal down a series of relay proteins. That's the signal transduction pathway. And the last relay protein actually changes that shape, allowing the cell to begin G1. Okay? As you can imagine, this is a super simple diagram, and that should leave you with tons of questions. Which means you get a 5 on the exam, you're going to take a cell biology class, and there will be entire chapters devoted to growth factors.
Whoa.
I know, right? How cool is that?
That's horrible.
Now, cancer, when good cells go bad. Okay? When good cells go bad, they don't pay attention. Those checkpoints don't work. Okay? And remember, we looked later on, we'll revisit cancer again when we looked at the genetic basis of cancer, and we talked about P53, the tumor suppressor gene. Okay? But at this point, you just need to know that cells are growing out of control. If cells are growing out of control, they form a tumor. If it stays in a solid mass, it's considered a benign tumor. Sometimes we don't do anything with it. Sometimes it can get too big. It can constrict blood vessels. And a lot of times we take them out because we don't want them to grow to become malignant. A malignant tumor starts sending out little fingers and little projections into the surrounding tissue. It also does something called angiogenesis. It stimulates the growth of more blood vessels to those cells because those cells need a ton of nutrients and a ton of energy because they're busy growing. And so it causes more blood vessels to grow near the tumor to feed the tumor. Ew, that's sort of creepy, huh? And then sometimes if we have a malignant tumor, metastasis can occur. Metastasis is where a bit of the tumor breaks off because they're no longer anchorage dependent, and then it floats through the body, through the bloodstream, through the lymph, until it lands somewhere else and starts forming a new tumor. So often one type of cancer can lead to another type of cancer. Okay? Question?
What happens when the blood vessels do come closer to the estimated towards the tumor?
Mm-hmm.
Are there any, other than the tumor growing larger and quicker, is there any byproduct of there being more blood vessels over there?
No, but it just means it's shunting more blood to the tumor and less blood to the other parts of the body, which means the other parts of the body aren't getting the nutrients that it needs. Okay? The last little bit of this section is 8.11. It's just sort of an overview. Why do we do mitosis, cell division, making identical daughter cells? We do it to grow, we do it to replace cells, and we do it for asexual reproduction. Well, we don't do asexual reproduction, but if we were asexual beings, we could do mitosis for asexual reproduction. So I'm going to stop this recording now, take any questions, and then we'll continue on with the meiosis part of this chapter. This is recording 5 of the second study party for the 2012 Christmas exam. And we are going to shift our focus for making a new cell a gamete. We do that by meiosis. Now that we understand mitosis, meiosis is sort of going to be a variation on the theme. The reason that we need to produce a haploid cell is so that we can have sexual recombination and keep the chromosome number the same. So, you usually have body you have body cells. They're called somatic cells. In humans, you have 46 chromosomes, 23 pairs. One of your 23 pairs came from mom, one of them came from dad. Okay? If you got all of mom's chromosomes and all of dad's chromosomes, you would have a different chromosome number.
Oh, that's a good idea.
You would be polyploid. Okay? And so we talk about diploid and haploid. Haploid, the haploid number is N. In humans, the N is 23. Diploid is 2N. 2 times 23, 46. Okay? So, the two chromosomes, you have two chromosome number ones, one from mom, one from dad. They are called homologous pairs. Homologous because they're similar, they have the same genes. Okay? The gene for tongue rolling or the gene for widow's peak or whatever, right? But they could be different because they could have different alleles. So they're homologous, but not necessarily identical. Okay? So, the place where a gene is located is called its locus. Okay? So, gametes only end up with a single set of chromosomes so that two gametes can fuse through fertilization and make a diploid zygote. Remember that a zygote is the single fertilized cell. As soon as we have a single fertilized cell, that single fertilized zygote is going to divide the rest of the time by mitosis, producing identical copies of itself. But even though they're identical, during development, we're going to permanently turn some genes off so that they differentiate and specialize. Right? Crazy, the story just gets more complicated. So, if we look on page 138 and 139, we see that meiosis reduces the chromosome number, and it happens in two steps, meiosis 1 and meiosis 2. Meiosis 1 is where the chromosome number gets reduced from diploid to haploid, because just like in mitosis, we have prophase. The chromosomes coil up, condense into the chromatin into chromosomes. But in this case, on the metaphase plate, the homologous pairs line up. And we call that a tetrad, for obvious reasons, right? In anaphase 1, the tetrad, the homologous pairs, separate, and the sister chromatids travel to opposite sides. And then we have cytokinesis. We don't need to go back to interphase because we've already replicated the DNA before we start. And then we have meiosis 2, which is essentially mitosis. In anaphase, the sister chromatids separate. And then we end up with 4 gametes. Okay? On page 140, in figure 8.15, they compare meiosis and mitosis. In many ways, it's similar, and it's super easy to get them confused. Okay? Now, it depends. We can line up those homologous pairs in any way we want. And so in figure 816 at the top of 141, we can see that we can produce, we're only looking at 2 different chromosomes here, right? But look at all the different possible combinations of gametes. So can you imagine how many different combinations we could get with 23 pairs of chromosomes? Holy cow. And then we know that homologous chromosomes carry different versions of genes, right? Big A, little a. But we can also have crossing over, section 818, which then totally increases genetic variability. The place where it crosses over is called a chiasma, which is the Greek word for X. Okay, and notice that we can have multiple chiasmas when the chromosomes are organized in a tetrad. Okay, so far so good. All right, and so now they're talking about crossing over, and they give you a whole step-by-step process in section 143. Okay, do remember that there is also in lab 7, in I forget which part of the lab, there was also, and we talked about crossing over frequency, and we could measure crossing over frequency with the Sordaria, and we looked at the crossing over rates with those little cards. Does that ring a bell? Black and tan patterns of crossing over. the ascospores.
With the black and tan. Oh yeah, I wrote that in here. Black and tan.
Yes, the black and tan fungi. Go back and look at that. Go back and revisit that lab, and I'll pull out the cards. I'll try and remember to pull out the cards for next class.
Did
If
we-
somebody wants to remind
Did
me.
we do it this year?
Is it an actual lab experience?
Well, no, it was sort of a dry lab. We looked at little photographs. We looked at little cards, and we did a cross, and then they stuck out in all different directions, and we were looking for black, 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 tan, tan,
I
tan.
Maybe
remember
as a
reading
volleyball
something.
or
No, I remember reading something about that, but I don't think we ever actually.
I don't know.
Maybe we didn't actually. Okay, so we'll take a look at that. Maybe that's one of the things we'll try to squeeze in on Friday or Monday. Okay? In section 819, we're looking at karyotyping. We try to get chromosomes that are in cell division so the chromosomes are all coiled up, and we look for what are called non-disjunctions. So where we have a trisomy or a monosomy. Okay? Down syndrome, trisomy 21. Okay? Very common. And so accidents that happen during meiosis can alter the chromosome numbers. Those are called non-disjunction events. So we end up with one too many chromosomes or one too few. That can happen in either meiosis 1 or meiosis 2. We can have non-disjunction events. And you can see on page 146, figure 821A and 821B, you can see the different results of what happens in non-disjunction and how different the gametes might look. Okay? If non-disjunction happens with the sex chromosomes, the X or the Y, it doesn't usually affect survival, where it truly can with other non-disjunction events on some of the autosomes as opposed to the sex chromosomes. And so you should know that XXY is Klinefelter's. You should know that XO is Turner syndrome. Okay, they can both lead normal lives. It does affect sterility, okay, and reproductive success. Okay, but if we have serious non-disjunction events, we can get whole new species, where if we don't undergo meiosis, we can end up with tetraploid, we can end up with polyploids, we can end up with many ends. Many of the fruits and vegetables that we eat are polyploid. That's what makes them so big and so juicy. Okay? Now, we can also have mistakes on the chromosomes. Section 8.24 on page 148. Okay? So, we can do several things. We can delete part of a chromosome. We're going to lose a bunch of genes. That would cause all kinds of issues. We can duplicate parts of a chromosome. Remember the ALU duplication? We're duplicating a chunk of the chromosome. Okay? We can have inversion. We can switch things around. We can have translocation. Often they're called reciprocal translocations, where parts of one chromosome switch with a non-homologous chromosome. And if you look down at 8.24c, you see a translocation between chromosome 9 and 22, and you end up with a Philadelphia chromosome, Philadelphia syndrome. Okay, there was a movie about that with Tom Hanks in it, I believe.
Is that Philadelphia syndrome or is that just AIDS? Because I'm confused.
Oh, maybe that movie he just had AIDS. Maybe that's it. Maybe I've got my movies all confused. He didn't have cancer, he had AIDS.
I
think he did.
I thought he
Oh, okay, then never mind. So it was a movie with the same name, but it wasn't the Philadelphia syndrome. So that we have this Philadelphia chromosome, which then causes chronic myeloid leukemia. Okay, and then the chromosome gets activated. So you have this cancer gene, but it doesn't get activated unless you have this reciprocal translocation mutation. Okay, so far so good?
So that's
Now,
unlikely
yes.
to have both the gene and then have
Right, but it happens. There's a statistical percentage of time that it happens. Okay? And that takes us to the end of chapter 8.